Good afternoon and welcome back. This week we are going to discuss how the location of transcription and translation impacts and differs between prokaryotes and eukaryotes. I saved y'all the pain of watching me draw all of this out as it was a slow process, but we will go through and label where the arrows are. So if you would like to go ahead and start copying down similar diagrams so that you have it to reference for your assessment at the end of the week, that would be advised. So within transcription for DNA, since there are multiple starting sites when beginning replication, this means it's bidirectional, which we have discussed. Therefore, there is a risk for it interfering with RNA polymerase. Because DNA conformations may impact the initiation, therefore the timing of replication and transcription, there's a chance for different elements to create traffic jams, overall increasing the chances of collisions and creating unnecessary genetic instability. For prokaryotes, since there's not a distinct difference where translation takes place, there is a risk for translation to begin before transcription has finished. So some cells may begin translation that have not undergone transcription, which creates an issue. Eukaryotes have introns and exons, which prevent this from happening. But back to prokaryotes, this process and the fact that it all happens in one location means that it's a simpler genome and there's less energy required. And it also means that the rate of translation affects the RNA structure, which then influences the rate of transcription. Eukaryotes, obviously, it takes place in two different places. Transcription takes place in the nucleus, and translation takes place in the cytosol, which we will label. And this means it's isolated, and also splicing helps increase the protein diversity and is also required for the mRNA molecule to be translated to then make a protein. So let's go ahead and label this chart that is already drawn out. So right here, we got our DNA. This arrow down here, we have our nascent proteins. Over here, we have our mRNA. Ooh. Cannot type. And then the purple is the ribosome. So the ribosome obviously makes the proteins after transcription and translation has occurred. The nascent proteins, which are the little pink dots, are proteins that are currently being formed. And then the mRNA is what carries our coding sequence. So over here we have our DNA still. Then this is our nucleus, which is where transcription takes place in this circle. Right here we have our primary transcript. And then right here begins the processing stage. So this is transcription is taking place up here, and then it goes, and then this is our mRNA strand. This is our cytosol, which is where translation is being taken place, which is occurring in this arrow, which is transport. It's transporting from the mRNA to the cytosol so that translation can take place. We have our proteins over here, and then we have our ribosome. So for the prokaryotes, the primary transcript, which is also this mRNA right here, this is used right after transcription occurs and goes right into translation, as this is a template for protein synthesis. So right after this happens, translation then starts, and then it goes and proteins start being synthesized through these ribosomes. So it's immediately. there's no processing and there's no transportation, it doesn't have to go anywhere, it's just immediate, which is why it doesn't take as much energy like we talked about earlier, and means it's a simpler genome. Then over here in eukaryotes, we have our primary transcript, and then it is processed into the mRNA. And then after the mRNA, it is then transported. This mRNA carries a coding sequence. It's transported to the cytosol, where then the mRNA is processed and spliced. Excuse me, I misspoke. mRNA is spliced and processed in the nucleus before being transported into the cytosol, which is where translation takes place. So after the mRNA is processed up here, it is then transported and translated into the proteins. All right, so thank you for watching. As always, if you have any questions, please feel free to reach out. Don't forget that your assessment is at the end of this week, so I highly suggest that you copy down this diagram and understand how the location impacts transcription and translation and how it differs between prokaryotes and eukaryotes.